Yep.
Go. Right. So this bit I want to talk about, carrying on from the triplet code, but discussing how a mutation in individual bases can cause a mutation. Now I'm going to do two sets of mutations, but these are called base mutations. Okay? And we need to be able to discuss them. And in particular, the examiner loves how a mutation could cause or could result in a non-functional protein. So we're going to try to do this now. Okay? We have read this mRNA code, and we have determined that that would mean that there are 4 amino acids in our polypeptide chain, and we would then put the peptide bond between them to form our polypeptide chain. And this would eventually be part of a large chain which would have a function, because it would be a protein. Okay? It would have a specific primary structure, which is the sequence and order of the amino acids, which would lead to a tertiary structure. If I can just step forward a bit, can we imagine that this combination of amino acids creates this structure due to the charges on both lysine and phenylalanine? All right? Can you see how the two sides, because of the positive and negative, are attracted towards each other, creating this shape that I've got there, this tertiary structure? Can we all see that? Okay? Now, this looks a little bit like something we talked about before. I'm going to try to say, can we imagine this might be an active site with a very specific shape that fits the substrate? Can you see that? Alright, now I want to go back and show what could happen. Because I want you to imagine, and there are set types of mutation. I'm going to do the first one first. The first one is a substitution. Alright? Now, I would like you to imagine, and we don't have to imagine, we're just going to do it, that at some point during DNA replication or in the translation or the transcription, of this mRNA strand, a mutation occurred and a different base was placed in a different part. And like a football, like when a player comes off and one comes on, we can call this a substitution, because I'm going to substitute the A for a C. Now, can you use your genetic code to tell me what the new codon, what amino acid it codes for? What amino acid does AAC code for?
Asparagine.
Right. ASP, you are telling me. Alright? Now that is going to change the primary structure just by that one letter change. Do you agree with that? Now that will mean that we will have a different amino acid here. ASP. And I'm going to tell you, and I'm making this up by the way, that actually ASP has a negative charge on its functional group. Can you tell me what that will do in this situation?
The repel.
These two are now going to repel. Will this affect the tertiary structure of this protein?
Yep.
Yes. What about if this was an active site? Will the enzyme now work? Right, now, this is what we need to do. We come up with a set points to talk about, okay? So, if the mutation could lead to a change in the primary structure, because you will change the amino acid, because the code's different. This change in the primary structure could affect the bonding. What are our 3 types of bonding in proteins?
Covalent, ionic, disulfide.
Ionic,
Covalent. Hydrogen.
and
Sulfide.
disulfide bridges. So it might be that the mutation changes the amino acid, which affects the bonding, which affects the tertiary structure, and this could lead to a non-functioning protein. Alright? And that is the whole set model answer for that part there. But I'd like to go back a little bit. I would like to perform another substitution. Again, a mutation occurs, a different base is substituted for an original base, but this time the A is substituted for a G. Can you tell me what effect that will have on the amino acid in that position? If you're looking and reading correctly, you'll realise that actually, due to the degenerative nature of the genetic code, actually AAA and AAG code for the same amino acid. Will that affect the structure of the protein? Will that lead to a non-functioning protein? Okay, so there's the two sides of the coin I need to talk about. That base mutations can lead to a non-functioning protein by the mechanism we talked about, and they also can have no effect. I just need to talk to you about some more. Alright? There aren't just substitutions where one base is swapped for another. There are insertions. There are deletions. Now these are what we refer to as frame shift mutations. And I want to go back to our original point here, our original diagram, and I want to talk about something. Can you imagine if, instead of substituting a base, I actually put a whole new one in? What will this now do to our strand of mRNA and our protein polypeptide chain?
To change all the amino acids after the first one.
After this first amino acid, we have shifted them all along one. So now I haven't got GGG, I've got AGG. And again, due to that non-overlapping nature of DNA, the genetic code, that's a completely different amino acid. I've now got GCC instead of what used to be CCC. Again, completely different amino acid. And frame shift mutations tend to be far more, I'm looking for the right word here, far more damaging, far more likely to produce a non-functional protein. And can you see why? Because we're not talking about individual amino acids here, we're having a whole frame shift. Okay? Good. Stop that for a second, Lewis, will you?